In this third video, what we're going to look at is just underscoring what we know from 2.7 and the process of transcription, and just adding one last level of detail about the regulation. So remember that we appreciate that transcription can be split into three sections. We've got initiation, elongation, and termination. Remember that initiation involves the binding of the RNA polymerase enzyme to the promoter region of the DNA. Remember that the promoter region of the DNA is interesting because it's non-coding DNA, and it's also subject to being regulated by enhancer sequences or silencer sequences, which are also non-coding DNA. And it's in order to regulate that, we need these transcription proteins that are involved. So if initiation is successful, RNA polymerase can bind, and what we then have is a copying of the RNA template strand to make a single-stranded RNA molecule. Remember that what we've got is an unraveling of the DNA at the level of the gene, not the whole entire chromosome, just the gene, RNA polymerase binding, and then that binding allowing us to produce this molecule of RNA. Now, this is referred to as being pre-messenger RNA, because what we now need to appreciate is that this then needs to undergo what's referred to as post-transcriptional modification. But here we've got the messenger RNA that's been sequenced, and remember that the messenger RNA sequence here, the base sequence, is going to be the same as this send strand at the top here. And also reminding you that this bottom strand, the anti-send strand, is acting as the template strand. Remember also that we've got three different types of RNA, but it's messenger RNA that we're interested in this particular instance. Here we've got a useful summary to underscore what happens during elongation. So we've got initiation, the binding of RNA, and then we've got elongation. And during elongation, we are using nucleoside triphosphates. Remember that we're breaking off the terminal two phosphates to produce the nucleotide, the ribose nucleotide. We're using complementary base pairing, cytosine always binding with guanine, and adenine binding with uracil on messenger RNA as opposed to thymine. So we got the sequence here on the send strand being similar to that on the messenger RNA. The messenger RNA is produced, remember, in the 5' to 3' direction, and it's an energetic process, which is why we've got those nucleoside triphosphates. That is going to happen all the way along until we hit a terminator sequence, and at the terminator sequence, the pre-messenger RNA will become disassociated with the gene, and the gene then can wrap itself back up again. Then what happens is post-transcriptional modification. Remember that on DNA we've got both introns and we've got exons. Exons are the coding sections of the gene. Coding sections referring to the fact that these are the sections which are ultimately going to be the code for the amino acid sequence during translation. Introns are the non-coding sequences which are taken out. So what we can see here is an exon, we can see the intron, and then we see another exon. So what we want to do is we want to cut out or splice out the introns, which are the non-coding regions. And to do that, we have what's referred to as a spliceosome. And what I need you to do here is to pause this video, go into our Google Classroom and play the McGraw-Hill animation, which shows you this very neatly. You don't need to know the level of detail that's there, but what you do need to appreciate is that what we've got is pre-messenger RNA, which then undergoes a process where we splice out introns to produce what's referred to as the mature messenger RNA. The spliceosome complex allows us to do that. What we then also have, which you can see here, is this idea that we can have a alternative splicing sequence. And this means that some introns can actually be referred to as being coding or non-coding, dependent on the timing of that. This also means that one gene can actually go on to produce more than one protein. Again, this is quite a new idea in biology. This is going beyond your syllabus, but the idea of splicing the messenger RNA means that the number of proteins that an organism can produce from one gene might differ each time. And that brings us to the end. Here is a reminder that you've got this big fat textbook. Please look through this. It does cover the higher units very, very well. And also BioNinja covers this quite nicely, especially in terms of using visuals. So what I'd like you to do now is go into the booklet for 7.2, work your way through that booklet and bring questions to the lesson for us to look at together.